let's look at the microscopic anatomy of a muscle cell. You can see from our picture here that each muscle cell is surrounded by endomysium. And imagine if we pulled one of those out like a straw and laid it down sideways. You can go ahead and use the green for that endomysium. Use the sharp pointy edge of your green highlighter if you can here. So there's endomysium, connective tissue, and then right inside of that there would also be the cell membrane of a muscle fiber, which is called the sarcolemma. And why don't we use an orange highlighter for that. And again, I'm using the pointy end to try not to take up too much room. So the orange is called the sarcolemma, and that's the cell membrane of a muscle cell. Muscle anatomists, that was one of the first areas that scientists were really able to study very in depth, and they loved all of the detail and specificity they were finding. So everything that was about muscles ended up with this sarco root on it. That root word, sarco, means flesh. And lemma means husk. So the sarcolemma literally means flesh husk. Interestingly, I bet you all have heard of this. A sarcophagus. So now you know sarco means flesh. You know that phagus means to eat. From general biology, at least, you've probably learned that. So sarcophagus means flesh eater. And what is a sarcophagus? It is a casket, usually one that a vampire lives in. Alright, so yeah, get used to that sarco root. You're going to see it a lot. Now, inside of each of the muscle cells are long organelles called myofibrils. I'm using a purple pen. Put in those myofibrils, and those are what I've represented with dots inside of the muscle cells. So each muscle cell is filled with myofibrils. Filled up to the brim with myofibrils. And in case you were wondering, these two circles here, I put those there to represent an artery and a vein traveling through the muscle. Okay, so purple is, those are the myofibrils. And they contain the contractile units that are called sarcomeres. There's usually about 2,000 sarcomeres in a single muscle fiber of the rectus femoris. So these sarcomeres are very tiny compared to the myofibrils. But a myofibril is composed of connected sarcomeres. Okay, now we'll go down and look at another microscopic view inside of a myofibril of the sarcomeres. These, let's use a green pen. I'm going to show you what's called the Z line, and the Z lines are what separate sarcomeres. So here's one sarcomere right there, and then here's another one, and here's another one. So in my little drawing, I've put 3 sarcomeres, and like I said, there might be 2,000 sarcomeres in one muscle cell, or one myofibril of a muscle cell. So green is the Z lines. And a sarcomere would be from one Z line to the other. And that word sarcomere means flesh, of course, and measure. So sarcomere is the measure of flesh. Now, within a sarcomere, you have actin and myosin, and those are going to overlap, and the myosin will pull on the actin to cause a contraction. The myosin is yellow, or not really yellow. I'm highlighting the myosin in yellow. This. And the way I've drawn this picture is with a relaxed muscle. If it were contracted, the actin would be, these two strands of actin would be as close together as they could. So, and see, then the whole thing would shorten. If it were an isotonic contraction. If it were an eccentric contraction, then the actin would actually be farther apart from each other. But you could see that the myosin has to be hanging onto the actin or the whole muscle will rip apart. So we went through and highlighted all of the actin in yellow. And actin is a thin muscle protein. And then we'll do myosin with pink. It's thick. So these are both considered myofilaments. Actin is usually called the thin filament, and myosin is called the thick filament. You can see that the myosin is a bulkier looking protein, and it has these little heads on it called myosin heads, and those are what stick to the actin and pull on it. So then in this one, I haven't drawn all of it out yet. Could put some myosin heads on there. Then in this one, start to get a feel for the arrangement of the actin and the myosin. So all of that pink that we did is the myosin. And myosin is not only thick, but it's also a motor protein, meaning it uses the energy of ATP to pull on the actin. In a relaxed muscle fiber, you can see a variety of areas, and if you were to look at them with a high-powered microscope, you would say, oh, that area is a little bit thicker. Look, it has actin and myosin in it. Let's call that the A band. So any area where myosin and actin are both overlapping is called an A band. So this would also be an A band. And anywhere that is just actin is called an I band. And anywhere where it's just myosin is called the H zone. If you were to look, though, at this under a microscope, then usually you don't see the H zone, because when the muscle is cut to prepare a slide, it contracts up, and the actin contracts together as closely as it can, and the H zone disappears. But a take-home message about these different zones and areas really what I want you to take away from this is understanding that when you learned that skeletal muscle has striations or stripes, those are caused by the careful organization of proteins inside of a muscle cell so that it can contract. Oops. So striations are caused and that's critical for contraction. So I want to go back to that idea of isotonic, isometric, and eccentric contraction. And let's see if I can do this very well. Let's, if you were to draw like this. Okay. And then put the myosin in this picture. The myosin has pulled the actin as close together as it can. So we would say this is what's happening in the most powerful isotonic contraction. And eccentric contraction is the opposite of that. You can see that most of the myosin heads are not able to even grasp onto the actin. They're hanging on for dear life, and if you were to stretch the muscle a whole lot more, then it would actually rip. So this would be an example of eccentric. But even though in this case the muscle is shortening and in this case the muscle is lengthening, in both cases the muscle is contracting and working quite hard. In fact, eccentric contractions will make you more sore because there will be small tears in different sarcomeres. And then an isometric contraction is sort of halfway in between, where it actually might look a little bit like this, where the myosin is contracting, trying to keep, but the actin is trying to pull away, but not being allowed to. But if you do allow it to stretch and contract at the same time, now you're looking at an eccentric contraction.